So this is just a short video. If you're not clear on the theories of replication, in order to understand, which are important for understanding Michelson and Stahl's experiment. So I'm just going to go through the three theories here, and then I'll refer back to them in the Michelson and Stahl video. So, but if you already understand them, then obviously you can skip this video. But if you're not sure, that's what this is for. Okay. So at the time of Michelson and Stahl's experiment, there were three main competing theories of DNA replication. Right. The first one is is called conservative, and it means fully conservative. Okay. And the idea for the conservative is that so now that they know the structure of DNA, they knew that it was this double-stranded helix, and so the idea is if this is the old strand, okay, that when it replicated, you would, or sorry, what this is the old molecule, okay. The idea is that the two new molecules that were formed, one of them would be entirely old, and one of them would be entirely new. Okay. So this is it, right? And so the idea is that it's conservative because both of the old strands are conserved together. And so the theory was that in some way these are being read and then producing a brand new molecule with two new strands. All right. So that's the conservative. The second theory is semi-conservative, and you guys should know that that is actually, in fact, how DNA replicates. It is semi-conservative, so I'll put a little star next to this one because it's the right one. Okay. And semi-conservative said that if you start out with one molecule with two old strands, right, then when you go to create two new molecules, each one of those would have one strand of old and one strand of new. And it would look like this. All right, so this is the correct one, obviously. And so in each one you have, so each molecule, you have one old strand and one new. And the reason why this works so beautifully is because of the complementary base pairing rule, right? The idea that A and T always go together and G and C always go together. This allows us to, so each of the old strands acts as a template for the new strand. Yeah. The other thing is I'm trying to be very careful in my wording, and you should as well when you answer these questions. So if you have two strands, if you have the double helix of DNA, if that's what you're talking about, that is one molecule of DNA. Each molecule of DNA has two strands of DNA, and I apologize, you can't really see the difference between the red and the blue on this, but hopefully there. And so the idea is that be very, very careful whether you're talking about strands versus molecules, right? The third theory was called dispersive. And so the idea for that one is, again, you have your old molecule with the two old strands, and then when you went to make new, the idea is that this basically got chopped up into little bits, and each little bit was then replicated and then assembled back together. Okay, so if I can try to draw that. Maybe I should pause for this part, but yeah, I've gotten a little off here, but okay. And so the idea is that each molecule, each molecule still has some old and some new. And for this first round, it would be 50% new and 50% old. Just like here, except rather than having one single strand be like one entirely new strand and one entirely old strand, the idea is that each little bit of the double helix has like this part is old and this part is new. But in terms of the overall percentage, it would still be the same. And this is important when we talk about Michelson and Stahl's experiment. Right. So those are the three methods that were floated in terms of how DNA could be replicated. And a lot of students think, well, these are stupid, and you think that because you know how it replicates already. It does this. the controversy around the semi-conservative method is that a lot of people thought that DNA strands could not be separated. And so the idea that there was some way to separate these two strands and then incorporate a new one, grow a new one basically, was what was controversial. Whereas these both they never involved the double helix being split open and the strands being separated. So that's why there were these different competing theories. But you don't need to know that for the test. You just need to understand what the three theories are and what they would result in. Okay.